Hello students, so welcome back to episode 22 of Decode NCERT, and in today's class we are going to decode the chapter biomolecules. So I'll be telling you, so before starting this class please sit with your NCERT book, your highlighter, one black pen, blue pen and a pencil so that you can note down all the things that I'm going to tell you. Now again I want to remind you the purpose of this video. It is not going to be a study video where I'll be teaching you the concepts, but yes, I'll be telling you what are the important things that you should not miss from this chapter. In different corners of this chapter there might be some important things that you may overlook because we are just focusing on important questions and all. So that you do not miss these things, I'll be telling you, so you have to highlight it. Okay, so very simple. Now let us move forward. Okay, so in the introduction part there is nothing much important, like you see in the amines chapter it was something very important things was there in the introduction part, but here it is nothing like that. Okay, next here something is called as what was the general formula of carbohydrates considered before. Okay, and then there was this is thing you should be knowing, that is hydrates of carbon. Next acetic acid, how does it fit here? We can write C2H2 whole 2. And then we have but it is not a carbohydrate, and similarly we have this example rhamnose. Okay, rhamnose which has a function formula of C6H12O5, which is not fitting to this formula but still has a carbohydrate nature and properties. So these two examples you should remember. Okay, next we have what is the definition of carbohydrates, this can be asked. Next it is the sugar that we use in our home are called as sucrose, that you should be knowing. Okay, and I hope many of you might be knowing it. Sugar present in milk is known as lactose. Carbohydrates are also called as saccharides. This you should be knowing. Why? Because in Greek saccharin means sugar. Okay, now we have the classification of carbohydrates on the basis of their hydrolysis. Okay, so first of all we have monosaccharides. So what is the definition of monosaccharides and how many monosaccharides are known? 20, and here are the examples. Now coming to oligosaccharides, definition of oligosaccharides, and then you have example, one molecule of sucrose on hydrolysis give one molecule of glucose and one molecule of fructose, whereas maltose gives only 2 molecules of glucose. These are the examples of disaccharides. Next we have polysaccharides, examples starch, cellulose, glycogen, gums, etc. And polysaccharides are not sweet in taste and hence they are also called as non-sugars. Okay. Now coming to another way how we can classify carbohydrates are your reducing and non-reducing sugars. So what are reducing sugars? Those who can react with Tollens reagent and Fellings reagent. Okay. Now we have something also monosaccharides. So classification of monosaccharides on the basis of functional group aldose and ketose if they contain aldehydes and ketones group. Okay. Next we have something about glucose. So where we find glucose, they are found in sweet fruits and honey and in ripe grapes. Okay. Now this is very important question for your PU board in the terms of like the structure of glucose, this you should be knowing. Okay, so this is very important. First one, from sucrose, let me zoom it. Okay, so the preparation is from sucrose, you should be knowing the reaction. Then you have from starch. Okay, that you have to remember, the preparation of glucose from starch is a commercial method, so you should be knowing this particular method. Now coming to the determining the structure of glucose, this is very important question. It can be asked as a 5-marker question, as a 2-marker question, as a 3-marker question. There are n probabilities of this question, so please study properly. So first one is your reaction with HI, the second one is your reaction with NH2OH and HCN, third one is the reaction of mild oxidizing agent like bromine. Okay, then you have reaction with acetic anhydride. Okay, and then you have here the last one, that is the reaction with nitric acid to prove that it has a primary alcohol group. So for all these reactions you should be knowing 2 things. Okay, so let me just note down here. So all these 6 reactions you should study like this. What is the reaction? Okay, and its conclusion. Okay, for everyone you should be knowing the reaction. Reaction you should be knowing in terms of statement plus in terms of reaction. Okay, and then what conclusion it gives, and here also you should be knowing the product. okay, and then what conclusion it gave you. Okay, this you should be knowing for all of them. Fine. Next, moving forward, we have here something is there, saccharic acid and gluconic acid structures also you should remember. Okay. Now moving forward, we have here what is D plus glucose. Why does it is named as D plus glucose? That is explained here. What does letter D and L stand, and its configuration with respect to glyceraldehyde, right. So this is so its configuration with glyceraldehyde, you should be knowing that if it is present on the right hand side we call it D glucose, if OH is present on the left hand side we call it L glucose. Okay. Next we have yes, that is all explained here, so you do not have to read much here, that is what you should be knowing. Next we can move here, there is nothing important. Okay. Next, having these are the limitations of the linear chain of glucose, right. So these limitations are very important, you should be knowing what are the limitations of open chain structure of glucose. Okay. Next we have the cyclic structure of glucose. Here you should be knowing about certain definitions, what is your anomeric carbon, that is the first thing, and after that you have anomers. Okay. That is there, what is anomers and what is anomeric carbon. Okay. Now coming to the Haworth structure, so you have to remember all these structures, and I know little confusing, so but yes, you have to prepare it every day and remember all the structures. Okay. Next we have fructose, where it is found in fruits, honeys and vegetables, and it is used as a natural sweetener. Okay. Now structure of fructose, it is also D and minus, it is levorotatory, so it is represented as D minus fructose, and it has a ketone group. Okay, and it is a 5-membered ring, and it is named, its name is similar to furanose, how it was in glucose, similar to pyranose. Okay. So you can see its alpha and beta structures as well, this is also you should be knowing. Okay. Then moving forward, we have come to disaccharides. So disaccharides, here what is glycosidic linkage, its definition. Then coming to here what is reducing and non-reducing sugars, that is there. Then now coming to sucrose, you should be knowing the linkage, what are the monosaccharides units and what is the linkage. So you have C1 of alpha D glucose and C2 of beta D fructose, these two are there. Okay, and it is a non-reducing sugars. Okay. Next we have sucrose is dextrorotatory, and here the concept of invert sugar is there, which is very important. Sucrose is itself a dextrorotatory, but when it undergo hydrolysis, it produces fructose and glucose, right. Fructose is levorotatory and glucose is dextrorotatory, and the fructose levorotatory is more than the dextrorotatory of glucose. So that is why the overall solution after hydrolysis becomes levorotatory, and that is what is called as invert sugar. Okay. Now we have maltose next. So what are the monomer units of maltose? So it is comprising of 2 alpha D glucose units, in which C1 of one and C4 of another combines. Okay, so you can see there, and it is a reducing sugar. Now coming to the third one, we have lactose, and in lactose it is known as milk sugar, as it is produced from milk. And then here it is comprising of beta D galactose and beta D glucose. Okay, and what is the connection? C1 to C4, and it is a reducing sugar because we have a free OH group here, right, which can be converted to aldehyde. Next we have polysaccharides. So starch is used as a storage polysaccharide in plants. Then where it is found? Cereals, roots, tubers and some vegetables. Then what are its type? You should know amylose and amylopectin, and the difference between both of them is given in this paragraph. Read these two paragraphs and make a table and write down the difference of amylose and amylopectin. Okay. Structure of polysaccharides are not needed. Structures of disaccharides are very important. Now coming to the cellulose, it is found in cellulose plant cell, and it is a straight chain of beta D glucose units which are joined by glycosidic linkage C1 to C4 linkage. Then glycosyl, it is an animal starch, structure is very much similar to amylopectin, and where it is found? Liver, muscles and brain, this is important. Okay. Next, it is also found in yeast and fungi. Now coming to importance of carbohydrates, nothing much is there. and we can move to now proteins. Okay. So, proteins are the most abundant biomolecules in the living system. Okay. In the living system, which is the most abundant biomolecule, it is protein. Okay. And where it is found? Milk, cheese, pulse, peanuts, fish, meat, etc. Now, protein has been derived from the word proteose. What is the meaning of that? Primary or prime importance. Okay. And all proteins are polymers of alpha amino acids. Next, we have amino acids can be of any type, alpha, beta, gamma, delta, but our proteins are made up of alpha amino acids. Okay. Next, what is how the naming of amino acids is done? It is basically done on the property that it's reflect. For example, glycine is named because it has a sweet taste, and in Greek glycos means sweet. Tyrosine was obtained from cheese, and in Greek again tyros means cheese, so it is tyrosine. So, that is what the naming has been done. Then it is represented by 3 letter and 1 letter, that no need to remember. Okay. Moving forward, here we have regarding classification of amino acid, basic and neutral amino acids. So, you should be knowing that if there is equal number of amino and acid group, it is neutral. If amino groups are more, then it is basic. If acidic groups are more, then it is acidic, right? And here we have also essential and non-essential amino acids, their definitions and examples of essential amino acids. Please do not forget, very important, please remember them. Okay. Next, we have zwitterion. So, what is a zwitterion? How does it exist? And you should be knowing this diagram for zwitterion as well. Okay. Fine. Next, coming to, we have, this is something important. Most naturally occurring amino acids have L configuration. L amino acids are represented by writing the NH2 group on the left hand side. Okay. So, amino groups are L configuration and how it is represented. Now, moving forward, we have the structure of proteins. Sorry, one second. Yeah, structure of proteins. So, they are linked by peptide bond, okay, and they are called as peptide linkage. And then we have this group, what I can say, the amino acids. Okay. So, we have here COH and NH2, there is a loss of water and a amide bond is formed, which is called as peptide bond, okay, CONH. Next, we have what is dipeptide, tripeptide, tetrapeptide, pentapeptide or hexapeptide, that you should be knowing. So, next, what is polypeptide? So, when there is more than 10 amino acids connected, it is called polypeptide, and here there is a very important definition of protein. So, a polypeptide with more than 100 amino acid residues having molecular mass higher than 10,000 U is called a protein. Okay. So, this is very typical, you should be remembering more than 100 amino acids and the weight should be approximately 10,000 atomic unit, then we will call it as a protein. But however, the distinction between a polypeptide and a protein is not very sharp. Polypeptides with fewer amino acids are likely to be called as proteins if they ordinarily have a well defined conformation of a protein. So, even though they have less amino acids, but if they have a conformation of protein, we can still call them as protein. For example, insulin, which has just 51 amino acids, it is not having more than 100 amino acids, but its structure is very similar to protein. Okay. Next, proteins can be classified into 2 types on the basis of their molecular shape. So, you know here fibrous protein and globular protein. Fibrous protein are held by hydrogen and disulfide bond, insoluble, and where they are found, this you should be knowing. Next, we have globular protein. So, these are found in insulin and albumins are the common example. Next, we have primary structure, okay, its definition, everything you should be knowing. Secondary structure, then you have alpha helix, okay, and then you have beta pleated structure. So, here there is nothing extra that you should be knowing, it is the same thing that is given in your every book, in your manuals, where the difference between alpha and beta. But something that is important here is okay, one second, yeah, tertiary structure. Then again in tertiary we have fibrous and globular proteins which stabilize the secondary and tertiary structures. These forces you should be knowing: hydrogen bond, disulfide bond, van der Waals and electrostatic forces of attraction. Okay. Now, quaternary structure of protein, we have here quaternary structures are referred to as subunits. Okay. And example, here are the structures, this diagram you can practice to draw in the exam, okay, primary, secondary, tertiary and quaternary. Next, we have something called as denaturation of protein. So, what is a native protein? First you should be knowing. Native protein is that which is found in the biological system with a unique 3-dimensional structure and biological activity. But when you disturb that protein, when you change the pH, temperature, and the protein loses its biological activity, that is called as denaturation of protein. Now, here if I tell you examples of denaturation of protein, you should be knowing egg white on boiling and curdling of milk, okay. Yes, next we have called as enzymes. So, what are enzymes? So, almost all the enzymes are globular proteins, okay, that is important. Next you should be knowing what is enzyme maltose does. It hydrolyzes, maltase enzyme hydrolyze maltose, okay. So, some enzyme and their function, then oxidoreductase enzyme, what do they do? Their function you should be knowing. Mechanism, here also we have an enzyme sucrase, it hydrolyze sucrose, okay. Next we have vitamins. So, what are vitamins? The definition you should be knowing. One second. Yeah, here it is the definition of vitamins, that is they are required in small amount but are very important for the growth, okay. Next we have here, that is something called as vitamine, okay. Why it was earlier called as vitamine? Because the word vital plus amine, and it was believed that vitamins are consisting of amino acids. But later it was found that vitamins do not contain amino acids. So, the E was dropped and we got the word vitamin. And in the classification of vitamin, we have fat soluble and water soluble. You should be knowing the example. Now, I will tell you this table is very, very important. It can be asked in MCQs, it can be asked in the 5-marker questions, it can even be asked in the 2-marker questions of biomolecules, okay. So, please do not miss this table. You should be knowing the chemical name, 2-3 sources, and diseases are very, very, very important. So, please remember the disease properly, okay. Okay, next coming to your nucleic acid. So, here I think nothing extra is there, whatever we have studied in the questions and all, the same thing is there. So, you should be knowing like, one second, yeah. So, what is the sugar present in RNA and DNA? It is beta D ribose in RNA and beta D 2 deoxyribose in DNA, okay. Structures no need to remember. Then we have what are the nitrogenous bases present in DNA and RNA, ATGC in DNA, AUGC in RNA, right. These structures also no need to remember. Then what is nucleoside and what is nucleotide, you should know the difference, okay. Next, next what is phosphodiester linkage, very important, should be knowing about phosphodiester linkage, okay. Next we have the functions of RNA and DNA, and here it is types of RNA is important, which is used in the protein synthesis part, okay. Now, moving forward, we have something that is your hormones, right. Yes. So, hormones definition you should be knowing, these are intracellular messengers. These are produced by endocrine glands in the body and are poured directly into the bloodstream, which transports them to the site of action, okay. Next I have something called as, one second, yes, here we have types of hormones and their function, okay. So, first is role of insulin to reduce the blood sugar level, then we have glucagon to increase the blood sugar level, then we have epinephrine and norepinephrine, which is used or responds to external stimuli. Thyroxine produced in the thyroid gland is an iodinated derivative of amino acid tyrosine, that you should be knowing. Next, low level of thyroxine leads to hypothyroidism. Low level leads to hypothyroidism and its consequences, or you can say symptoms are letharginess and obesity. And if thyroxine increases in the body, it releases, it is the condition of hyperthyroidism, and we have hypothyroidism and enlargement of thyroid gland takes place in that case, and how it is cured by iodized salt, okay. Then we have steroids hormones, that is your adrenal cortex and gonads. So, which are your testes in males and ovaries in females. Next we have glucocorticoids that control the carbohydrate metabolism. So, this whole paragraph is very important, okay, where I have given you all the functions of the hormone. So, you have to read it, okay, right. In my case set biomolecules chapter, if you go, I have given this data in tabular form, the hormones type and its function. So please go to that video, and I will be putting the link of that video in this description section. So please go there. Once you read, just go to that video and make the table and finish it. Okay. And one more important thing, something which is hidden here is your addition disease. You should be knowing what is addition disease and when does it happen. It happens if your adrenal cortex does not function properly. Okay. And it is, what is the consequences of that? We get hypoglycemia, weakness and increased susceptibility to stress. Okay. Then here we have the male and female sex hormones, their functions and their importance that is given, and that is all. Okay. So, yes students, I think this chapter is over. Okay. So, yes, so in this chapter I will say nothing, there is much which is hidden from you. Like hidden means we generally encounter all the concepts while solving the basic questions of this chapter. Okay. So, this is a must do chapter, easy and scoring topic. So, make sure that you study all of them, and if you have any doubt in any concept, do comment down, I will definitely address. Okay. So, thank you students, and please let us know how was the series, like whether you are enjoying this Decode NCERT series that we have started, and do you want any extra thing in this? So, please comment down, we will definitely address. So, that is all students, and thank you, and please do not forget to subscribe the channel, and see you soon in the next class.